Dear, dear students, today we'll talk about a very important topic, that is jaundice. Okay, this is very important. Okay, so here you can see the image. Okay, so by this image we can tell that there is change in the conjunctiva part of the eye, and you can also see that there is yellowish hue in the skin. Okay, and it's very pale in color, so the skin is very pale yellowish color of the conjunctiva. Okay, so by seeing we can see this yellowish discoloration of the skin. Now clinically, by definition, what is jaundice? So jaundice other name is icterus. So what does it mean? It refers to the yellow appearance of the skin. As we said, as we have seen in this picture, yellowish discoloration of the skin, and we can see that the conjunctiva of the eye has also turned yellow in color. So jaundice, other name icterus. Okay, it refers to the yellow appearance of the skin, sclera and mucous membrane. Okay, now what is the reason behind this yellowish discoloration of the skin? So the reason behind is increased bilirubin level in the body. Also we can say hyperbilirubinemia. Okay, now clinically here means we can test the level of bilirubin, which is responsible for development of this yellowish discoloration of the skin. Clinically, jaundice is detected when the plasma bilirubin level exceeds 2 to 3 mg per deciliter. Okay, so in the upcoming slides, we'll see what is the detectable plasma, what is bilirubin. Okay, we'll see in the next slide. Now, so jaundice or icterus, which means yellowish discoloration of the skin. Now we'll see what are the types of jaundice. So, prehepatic jaundice. Prehepatic jaundice, it means that in this condition, what happens? There is excessive red cell breakdown. Okay, which the liver is not able to conjugate the bilirubin, means here there is excess production of the bilirubin. See, after 120 days what happens, that our RBC will destroy, and ultimately we get bilirubin and biliverdin. Okay, now what happened, this bilirubin has to be conjugated by the liver, but when the production of bilirubin is very high, the liver is not in a situation to conjugate this bilirubin. So this causes the level of bilirubin to exceed in our body fluid. Now, this is the prehepatic jaundice is also, we can tell that it is unconjugated type of hyperbilirubinemia. So in this condition, where the liver inability to conjugate the bilirubin, and ultimately it is unconjugated hyperbilirubinemia. Okay, now bilirubin, actually what happens, bilirubin will become conjugated with the liver and it get excreted from the body. Okay, now this unconjugated bilirubin, it remains in the bloodstream and it causes the jaundice. Jaundice over here means that the level of bilirubin is increased and it is detected in our body fluids. Next type of jaundice is hepatocellular or intrahepatic jaundice. So here what happens, intrahepatic means there is a problem within the liver itself. Means here there is dysfunction at the level of the liver. Means what happens here, there is problem in the hepatic cells. Actually what happens, the liver loses the ability to conjugate the bilirubin. Okay, and it what happens so there is problem with the function of the liver, and so the condition which is developed is jaundice, but the problem here is at the level of liver, so it is called as intrahepatic jaundice or hepatocellular. Actually what happens in some cases, in cirrhosis, what happens, the intrahepatic portion of the liver, it becomes abnormal. Okay, so the biliary tree will get obstructed, so that also causes problem. Next, post-hepatic jaundice. So post-hepatic jaundice refers to obstruction of biliary drainage. So there is a problem at the level of drainage. Okay. So the bilirubin which is not excreted will be that result in conjugated type of hyperbilirubinemia. Now, for our better understanding, we'll see what is the fate of our RBC and mechanism of liver for conjugation of bilirubin. Actually, what happens is, okay. See, uh, this hemoglobin, after destruction of RBC, the hemoglobin part will disintegrate into two part, that is globin and heme part. Okay. Now this globin will enter into amino acid pool, but this heme part will again be reused for the synthesis of hemoglobin, and it will split into iron. Okay, and the remaining part will get oxidized by oxygenase, and then we get biliverdin and biliverdin. Again this biliverdin, it will be reduced by an enzyme, that is biliverdin reductase, to get bilirubin. Now this iron part will combine with epiferritin, and we get ferritin which gets stored in the liver. Okay, now this bilirubin, they are excreted in the bile. Okay, actually what happens now, this bilirubin is actually what is produced. They are unconjugated bilirubin. These are lipid soluble. In the plasma, it is bound to the albumin. When it is bound to albumin, it is called protein conjugated, which will prevents it excretion by the kidneys. Okay. Now in this diagram also we can see that this RBC, it has got after 120 days, it has been destroyed. Okay. And then this bilirubin, now this bilirubin, it will bind with glucuronic acid, which is called conjugated bilirubin. Okay. Now what happens, this bilirubin will again, when it comes, this bilirubin by the intestinal enzyme beta glucuronidase, there will be we get glucuronic acid and urobilinogen. Okay, and finally we get stercobilin, which is excreted in the fecal matter. 20% of urobilinogen will again will enter into enterohepatic circulation. Okay. Now we'll see what is conjugation of bilirubin. The unconjugated bilirubin means, which is from the circulation, is taken up by the liver. As we have seen in the diagram, it is taken up by the liver. In the liver, the bilirubin it will split from the albumin, and it will enter into the hepatic cell. In the hepatic cell, it is conjugated with uridine diphosphate glucuronic acid, making it water soluble conjugated bilirubin. This reaction of conjugation of bilirubin with UDP glucuronic acid, it is catalyzed by the enzyme glucuronyl transferase, which is present in the hepatic microsomes, okay, which is smooth endoplasmic reticulum of liver cells. Now, excretion of bilirubin. Now, what happens? The conjugated bilirubin from the hepatic cell, it is excreted into the bile and enters the intestine. Some of it escapes into the general circulation and is excreted by the kidney in urine as urine bilirubin. This is what the diagram we are talking about. Some of them, some goes into the liver and other they come into the kidney and they are excreted as urobilinogen. Okay. Now we'll see what is formation and excretion of urobilinogen. The conjugated bilirubin which enters the intestine, it is degraded by the intestinal bacteria in the terminal ileum and the large intestine. The bacterial enzyme beta glucuronidase splits off the glucuronide and converts bilirubin into urobilinogen. So again, this diagram. So this bilirubin, again it comes into our intestinal circulation. When it comes into intestine, it is activated. It is degraded by bacteria of the intestinal flora, and with the help of the enzyme which is beta glucuronidase, and we get glucuronic acid and urobilinogen. Okay. Now, some urobilinogen from the intestine is reabsorbed and goes via the portal system to the liver. From the liver, some urobilinogen escapes into general circulation and some are re-excreted into the bile, which is also known as intrahepatic circulation. So here we can see the 80% it is comes out as stercobilin and remaining 20% it enters into the enterohepatic circulation, which can be reused. Now, from the general circulation, the urobilinogen is filtered off by the kidney and is excreted in the urine. So, the remaining 80% of stercobilinogen in the intestine, which is not absorbed, is excreted in the fecal matter, which is approximately 20 to 250 milligram per day. This stercobilinogen is oxidized to stercobilin, which gives fecal its brownish color. So this portion. Now this RBC, which is degraded into bilirubin. Now bilirubin, it binds with albumin. Now this bilirubin, when it comes, it binds with the glucuronic acid and it is called as conjugated bilirubin. Out of this, 80% will get excreted in the form of stercobilin and 20% will enter into enterohepatic circulation. Now we'll talk about bilirubin. Bilirubin, the normal serum bilirubin level, it ranges from 0.3 to 1 milligram per deciliter. The total serum bilirubin includes conjugated as well as unconjugated bilirubin. So the test, see, as we have already mentioned in the previous slide that clinically it can be detected. So by which test we can detect? So the test which can detect this bilirubin in our serum is Vandenberg test. So it helps in the detecting of bilirubin in the serum. Okay, so Vandenberg test is performed using the diazo reagent, which is mixture of sulfanic acid, hydrochloric acid, and sodium nitrate. It is of two type: direct Vandenberg test and indirect Vandenberg test. So this was all about your jaundice and type of jaundice.